Boy Littleton, it's my pleasure to introduce this year's Elkins Memorial Lecture. The Elkins Lecture was established in 1991 to honor the memory of Tom Elkins. Tom earned his PhD with Barry Ganetsky in 1986. His groundbreaking electrophysiological characterization of the slowpoke mutant demonstrated that it lacked a calcium-activated potassium current, focusing attention on slow as the structural gene for these channels. Subsequent cloning of the slow locus led to the first molecular analysis of calcium-activated potassium channels. Following his PhD in Barry's lab, Tom moved to Corey Goodman's lab for postdoctoral studies, and he carried out the first detailed genetic analysis of neural cell adhesion molecules by generating and characterizing mutations in the fasciculin gene family. Tragically, Tom was killed by a drunk driver during his postdoc, and his mentor and friends were notified of his death during the Cold Spring Harbor meeting that year. The Drosophila community joined together to collectively remember Tom, and Corey Goodman and Barry Ganetsky proposed the creation of the Elkins Lecture to recognize outstanding achievement by a graduate student in the field of Drosophila neurobiology. The first Elkins Lecture was held in 1991, and it's become a highlight of the Drosophila meeting, as many of you know, since its establishment more than 30 years ago. By recognizing the accomplishments of outstanding young members in our field, the lecture series has provided a lasting memorial to celebrate Tom's life. So the Elkins committee had a difficult task of picking a winner and an honorable mention from an excellent group of applicants. And I just want to congratulate all the nominees and their advisors. They should be very proud of their work. This year's honorable mention is being awarded to Ellen Lesser from the University of Washington, who did beautiful work in John Tuthill's lab to provide insights into the structure and function of sensory motor circuits that control Drosophila flight. The winner of this year's Elkins Memorial Award and our lecturer for this evening is Matthew Capek. Matthew completed his graduate thesis work in Marco Gallio's lab at Northwestern, and Matthew's research has provided fundamental insights into how the brain processes external temperature to regulate innate behaviors, as well as how species-specific temperature preferences are established. Congratulations, Matthew. We're excited to hear about your thesis work.
All right. Hello, everyone. Thank you so much for this recognition. It's really great to be here honoring Tom Elkins' memory, and also to tell you about the research that I've done during my PhD in Marco Gallio's lab at Northwestern. So temperature is a really important environmental variable that has a fundamental impact on biological systems. And as such, the ability to seek out optimal temperatures in the environment and avoid potentially hazardous ones is a really important behavior for all modal animals. Take, for example, these three different fox species that are closely related, so in the same genus, but inhabit very, very different thermal environments, from the harsh cold of the Arctic to the extreme heat of the Sahara Desert. And so this adaptation to these different environments is usually thought of as a coordinated set of changes in biochemistry and physiology that make it possible for these species to tolerate these different temperatures and then eventually thrive in these habitats. But importantly, they must also align their preference for temperature with the conditions of their habitat, sort of as the optimum temperature for survival changes as the species adapts to a new range, right? However, we know little about how animal behavior evolves in general. We're really only scratching the surface. And we know even less about how temperature preference evolves as an animal adapts to a new environment. And so to address this question, we use species of Drosophila, of course. And so Drosophila flies are not only really great models for this because of the extensive genetic and experimental toolkit we have in Drosophila melanogaster, but in fact because there are over 1,000 different species of Drosophila that are found all over the world and in basically any climate you can think of. So they represent this really great model genus to address how temperature preference has evolved. So first I'm going to give you a brief introduction to the thermosensory system in Drosophila. So in order to sense external temperature, Drosophila use their antennae. And so housed within this feather-like structure called the arista here are the thermosensory receptor neurons, where in each arista there are 3 distinct neurons that respond to cooling, called cold cells, and 3 that respond to heating, called hot cells. So here this is calcium imaging, where you can see two hot cells here activated during a heat stimulus, and there's only 4 visible in this focal plane, whereas the cold cells are activated only by cold temperature. And these sensory neurons then send their axon into the brain through the antennal nerve and terminate in this region called the posterior antennal lobe, or PAL for short. and they form distinct but adjacent glomeruli for hot and cold respectively. So it forms this really simple sensory map for the representation of external conditions. From there, then thermosensory projection neurons then receive input from the PAL and target higher order processing centers in the brain, such as the mushroom body, the lateral horn, and the posterolateral protocerebrum. And the Galileo lab, among other labs in the field, have characterized a variety of different TPN subtypes with different connectivity, anatomies, and firing properties. So for example, some TPNs actually receive input exclusively from just the cold cells, like this one I'm showing you here. So it has dendrites only in the cold glomerulus, and it has this interesting anatomy where it targets the lateral accessory calyx, which is actually a known center for sleep and circadian regulation of behavior. Whereas others, like this one here, receives input exclusively from hot cells, and they have diverse anatomies and connectivity. And then finally, some are what we call broadly tuned, and so they receive input both from hot and cold. So for example, this one actually innervates all three of these different brain regions. And at the molecular level, compared to mammals, the receptors expressed in the sensory neurons in the fly that directly mediate sensing of temperature, actually partly conserved and partly unique to the fly. And so in Drosophila, the receptor for innocuous hot is this ion channel called GR28BD. For cold, the candidate cold receptors we have are IR channels, so IR21A and 25A. And then for sensing of noxious heat and other noxious stimuli, there are TRP channels like TRPA1. And of course, TRPA1 is very highly conserved among animals. Okay, so how then has temperature preference evolved in different Drosophila species that have adapted to these different climates? And what components of this thermosensory system have acted as sort of the evolutionary substrates for this behavior? So to measure temperature preference of flies in the lab, we use this assay called two-choice assay, where we place flies in these circular arenas, where we give them a choice between a base temperature, which we use 25, and then a range of different test temperatures. And so we do this by having these floor tiles where we can individually set the temperature in these 4 quadrants. And so we can count the number of flies and compute an index, so like a preference index, where +1 means the flies completely avoid the test temperature, whereas -1 is the opposite. So in particular, I chose to focus on 3 species to address this question that inhabit different environments. So first, of course, is Drosophila melanogaster, which actually originated in sub-Saharan Africa, but is now found all over the world following human populations. And so consistent with this, actually melanogaster prefers 25 degrees as its favorite temperature. And so we see that in this assay, Drosophila melanogaster will very robustly avoid temperatures that you show to them that are above 25 or below 25. And then next is Drosophila persimilis, which is a species that's restricted to the coasts of the Pacific Northwest. So these are found in sort of the cold coniferous forests in the Sierra Nevada range of California, for example. And so in line with its natural environment, this species prefers distinctly cooler conditions than melanogaster. And so it prefers the test temperatures of 15 and 20 as its favorite temperature, and will avoid any temperature above 20 degrees. And finally is Drosophila mojavensis, which is my favorite Drosophila species. It's a fly that's endemic to the deserts of southwestern United States and Mexico. And it's actually the most heat-tolerant Drosophila species. And it can withstand temperatures reaching upwards of 50 degrees. And in fact, mojavensis actually likes it hot, surprisingly. And so they actually prefer 30 to 35 degrees as their preferred temperature. And so this is really striking, right? Because these are temperatures that are very strongly avoided by the other 2 species. So overall, what we find is that these species actually display specific preferences for temperature that sort of correlate with the conditions of their native habitat. So as I introduced earlier, we know actually sensing of innocuous heat is mediated by this ion channel called GR28BD. And these are expressed exclusively in the hot cells in the arista that you're seeing here, labeled with GFP. And this here is actually an alpha-fold structure prediction of GR28BD. So it forms this homotetrameric ion channel with a predicted pore that looks very similar to the solved structure of like Orco, since it belongs to the same sort of superfamily of ion channels. And an interesting sort of side note that is really Peculiar is that the GR28B locus is quite interesting and complex, so there actually produces 5 different gene products, starting from this nested set of promoters. So each isoform has its own independent start codon, and then they're all spliced with these common exons here at the end. And so only this D variant is the thermosensory one, whereas these other variants of GR28BD are actually expressed in the taste system, and some of them have been characterized as bitter receptors. And so this GR28BD protein is likely evolved via, its thermosensitivity is likely evolved through duplication of one of these chemosensory variants, and then subsequent acquisition of its thermosensitive function. Okay, and so we know that GR28BD actually functions as a heat receptor in melanogaster because unlike wild-type flies, where if you give them a choice between 25 and innocuous heat at 30 degrees and painful heat at 40, we see that compared to wild-type, the GR28BD mutants completely lose avoidance of innocuous heat, whereas they still avoid noxious heat at 40 degrees just fine. And this is because sensing of noxious heat is mediated by TRPA1, so through a separate pathway. And so if we actually knock out both GR28BD and TRPA1 in the same fly, you can make a fly that's completely heat blind. And so these flies actually just run around at 40 C and then just die. And so by recording their activity in culture cells, GR28BD is actually directly gated by heat. And so compared to this control cell here, if we express this ion channel in cell culture and do patch clamp recordings, we see that the melanogaster GR28BD, we see these robust inward currents in response to heating. And it turns out that this activation is actually characterized by a specific threshold that we can measure from this data. And so we use this Arrhenius plot where we can essentially look at how the current is changing as a function of temperature. And across multiple recordings, what we find is that the threshold of activation for melanogaster GR28BD is actually 26 degrees. And so looking back at the behavior of melanogaster, this actually makes a lot of sense because this nicely sort of corresponds to the onset of heat avoidance behavior in the fly, right? And so one hypothesis we had is that maybe these 3 species I just showed you before may have differences in their GR28BD threshold that could explain these sort of shifts in heat avoidance behavior. So for example, maybe Persimolis has a lower threshold that sort of explains why it avoids heat at a lower temperature, whereas Movensis maybe has a higher threshold of activation. And my results sort of partially support this idea. So first, compared to Drosophila melanogaster, the GR28BD threshold of Persimolis is indeed lower, approximately about 21 degrees on average. And so this corresponds to the lower onset of heat avoidance for the species, which only start to avoid heat above 20 degrees, right? And so as temperature is increasing, just above the preferred temperature, this receptor gets activated, and then the flies know to avoid. And second, Movensis indeed actually has a higher threshold of activation at 29 degrees, the highest of these 3, which correlates with the hotter conditions of its environment. However, this threshold really sort of puzzled us at first because it actually doesn't correspond to heat avoidance at all, right? Because Movensis actually prefers 30 to 35, so it's just below their preferred temperature. And so if this shift in behavior across species was fully explained by just shifts of the threshold of this receptor, we would instead expect the threshold to be above 35, right? So above their preferred temperature when they actually start to avoid heat. And so this suggested to us this remarkable possibility that something else could be going on, right? So maybe activation of the hot sensing neurons in Movensis doesn't mediate aversion like we know it does in melanogaster, but instead maybe the valence of the signal is actually switched in the brain, and maybe they're actually actively seeking out heat. And so to test this idea, I carried out this set of gene swap experiments of this GR28BD receptor between species. And so here I'm showing you the same 2-choice preference assay that I showed you before, but I've also added this test temperature of 27.5 to better discriminate differences in threshold. And so you can see that wild-type flies will avoid heat above 25, but the CRISPR mutant for GR28BD, these flies lose avoidance of 27.5 and 30, so innocuous heat. And this can be rescued by expressing back in the receptor in the hot cells, and so we see this completely restores wild-type avoidance behavior. But if instead I express the mohavensis ortholog of this channel in the hot cells of melanogaster, so this cross-species rescue, as you remember, the channel from mohavensis actually has a higher threshold. We indeed rescue heat avoidance in melanogaster, but we only rescue avoidance of 30 degrees and not 27.5. And so effectively what's happened here is we've rescued heat avoidance but imparted a mohavensis-like threshold to the behavior. Okay. And so what about doing this gene swap experiment in mohavensis? And so mohavensis, of course, prefer 30 and 35 degrees as a preferred temperature, and they actually have no preference for 27.5. And we created a CRISPR mutant in mohavensis for GR28BD, and indeed we see these flies lose attraction to heat. And so now if we actually rescue this mutant in mohavensis by expressing the melanogaster ortholog of GR28BD in the hot cells, which, as you remember, has a lower threshold than is in the endogenous GR28BD, we indeed rescue heat attraction in this fly, but we even see attraction to 27.5, which is a temperature that wild-type mohavensis flies are completely indifferent to. And so effectively, what we've done here is we've rescued the behavior in this species, but imparted, again, the threshold from the donor species. And so this gene swap experiment supports this notion that GR28BD is required for responses to innocuous heat in both of these species. But this signal actually carries the opposing valence in melanogaster and mohavensis, where activation of GR28BD mediates aversion in melanogaster and attraction in mohavensis. And so what is then different between these two species that could explain this switch in valence? And so are there any differences then in the central circuit in the brain that's processing this heat information in mohavensis that could explain this switch in valence? And so in other words, has the thermosensory brain circuit been rewired across evolution? And so to begin exploring this, I first created some transgenics in mohavensis that express GFP under the GR28BD promoter. And so we can see that mohavensis indeed has 3 hot cells that look similar in anatomy to what we see in melanogaster. And following these projections into the brain, they both project to the posterior antennal lobe in essentially the same region. They look very similar. And so now that it looks like we've confirmed that the sensory neurons, or first-order neurons in this circuit, are conserved in mohavensis and melanogaster, the next sort of logical step would then to be what the projection neurons look like that are relaying this information to sensory processing centers. And so these TPNs, the thermosensory projection neurons that I talked about earlier. And so in order to label the TPNs in the mohavensis brain, I adapted this technique for circuit mapping called electroporation. And so using this transgenic that I showed you before that labels hot cells, I targeted an electrode that's filled with a dextran conjugated dye to the axon terminals of the hot cells in the brain under a 2-photon microscope. And so then following a sequence of electrical shocks, this creates little pores in the membrane and allows the dye to fill the cell. And so then this dye, intercellular dye, can then fill the neuron and we can visualize its anatomy. And it's worth noting that this technique actually builds from pioneering work from Ed Kravitz, who was one of the first to show using intercellular dyes for visualizing neuronal anatomy. And so with this technique, I can then be able to stochastically label these TPNs that innervate the hot glomerulus in mohavensis. And so what I found overall is that it seems like the gross TPN pathways in the brain are conserved in mohavensis. And so on the left here are examples of well-characterized TPN cell types from the Drosophila connectome here, and then on the right is these neurons that have been filled with a dye in mohavensis with a 2-photon microscope. So you can see their morphologies and anatomies in the brain look very similar. However, of all the neurons that I labeled, this TPN that we call TPN5 really stood out, and this is for a couple of reasons. So this is because we know from the melanogaster connectome data set that TPN5 is actually the primary output of the hot cells in the brain in melanogaster. And so it comprises roughly 26% of the of the output from the hot cells. And in my dye filling experiments, although I did label these other TPNs, TPN5 was by far the most frequent one that I labeled in these experiments. And so approximately 80% of the brains that I did electroporation with, TPN5 was filled. So I think this is kind of owing to its really dense innervation in the hot glomerulus. Okay, and so in melanogaster, TPN5 innervates two different brain regions, so the calyx of the mushroom body and the lateral horn. And so it passes laterally to innervate the calyx. It has some synapses in the main volume of the calyx, but then it has this sort of more prominent branch in the lateral accessory calyx, which, as I said before, is actually a center for regulation of circadian behavior in sleep. And then it sort of veers more ventrally to innervate this sort of ventral portion of the lateral horn. And in dye filling experiments, I can capture the same anatomy from the connectome in melanogaster. Everything looks great. But finally, this is what actually TPN5 in movensis looks like. And this was very exciting because we can actually notice some interesting differences in its anatomy. And so it actually has a slightly more prominent and longer branch in the lateral accessory calyx. We don't really fully understand what's going on there, but it could hint at maybe some differences in temperature regulation of sleep. And there also, we notice a difference in the lateral horn as well, denoted by this arrow. But this difference in the lateral horn is much more clearly seen if I flip the brain 90 degrees and image from the dorsal point of view. So if I do that and image the brain dorsally, we see in melanogaster, this is what it looks like. So on the top here is a representative two-photon image. And then here I've did 3D reconstructions of these neurons that have been registered to the same volume. So this is n equals 5 neurons. And so you can see in melanogaster, TPN5 sort of comes in at this angle, and it has an extensive branching that goes out sort of in the center of the lateral horn. But in movensis, there's quite a striking difference. And so it actually, its innervation is restricted to the posterior domain of the lateral horn. And you can see it actually has a more of a flat angle, and it doesn't have these sort of more extensive branches that are seen in melanogaster. And this was really exciting because this suggests that there might be differential connectivity of this neuron in movensis. And since this region is actually known to be really important for processing of innate valence of sensory information. Okay, and so you may be wondering like how melanogaster and movensis are related. And so they've actually evolved separately for roughly 40 million years, so they're quite diverged. And so one sort of potential explanation for this is maybe this different pattern of innervation of TPN5 could just reflect sort of gross organization of the lateral horn. Maybe things have just been rearranged across evolution. And so to explore this possibility, I wanted to be able to label a well-characterized projection neuron from the olfactory system that also innervates the lateral horn sort of as an internal control in the same brain. And so I labeled a brain in movensis with two different colors of dye using this technique. One that actually labeled TPN5, and then one that labeled this projection neuron from the olfactory system, DM1. And so in melanogaster, in both the EM reconstruction on the left and the two-photon images on the right, you can see that these two projection neurons actually sort of overlap in the center of the lateral horn and actually have the same sort of angle of entry into the lateral horn. Whereas in movensis, you can see that TPN5 has a distinct sort of projection. So it has this more posterior pathway, and you can see it's with this internal control, we can see very much so a clear lack of innervation in the interior lateral horn. And so these results demonstrate that we can potentially sort of likely rule out the possibility that the lateral horn has been completely rearranged across evolution. Okay. And so what I've shown you thus far is that melanogaster and movensis have a significant difference in the anatomy of this projection neuron. That's really a key conduit of relaying heat information from the periphery to the central brain. And so this is all well and good, but in further support of this, Drosophila persimilis, which you remember from earlier, also avoids heat like melanogaster. We see that if I label TPN5 in persimilis, its innervation of TPN5 in the interior lateral horn is much more close to melanogaster than movensis. However, if you look at this tree, persimilis is also quite separated from both of these species in terms of evolutionary time, and so this could also just reflect phylogenetic relationships in these different species groups. And so to directly test this, we were very lucky to find this sort of natural control species to moavensis. So this is actually a species called Drosophila metleri. And so this is another desert fly that's very closely related to moavensis. But unlike moavensis, it actually avoids heat. So we can see here it actually avoids heat above 27.5, and it actually has a threshold of GR28b that, again, nicely aligns with the onset of heat avoidance. And lo and behold, sort of despite being closely related to moavensis, it has clear innervation in the interior lateral horn that more closely resembles that of the other heat aversive species, despite being closely related to this other sort of sister species of moavensis. But this is only one brain, and so I wanted to be able to sort of visualize these differences in anatomy in a more quantitative manner. And so I use this pipeline where I take these 2-photon microscopy images, segment the brain, and register them to the same volume within species. And then I did tracing of the neuronal anatomy to sort of get a 3D reconstruction of their branching structures. And so with these reconstructions in hand, I first computed the sort of the pairwise differences in branching anatomy of these neurons, which tells us about their sort of intrinsic morphological differences. And then also use this measurement of center of mass, which tells us about the spatial innervation of the neurons in these brain regions. And then finally, I use this hierarchical clustering model to integrate these two different metrics, and then using dendrogram and UMAP to visualize differences between the structures. And so what we see is that actually the innervation of the lateral horn is significantly different in moavensis compared to the other heat aversive species. And so you can see in these n=5 of these reconstructions above, but also here in this hierarchical clustering, we see that the moavensis TPN5 neurons cluster distinctly from the other heat aversive species. You can see this also represented in the dendrogram. And as in control, we really don't see any distinct clustering in the calyx, where all the species, the structures in this multidimensional space sort of tend to cluster all together. And so in conclusion, I've shown you that in Drosophila, the adaptation to different thermal niches has involved changes in thermal preference that sort of reflect the conditions of their natural habitats. And we've identified two distinct neurobiological mechanisms that drive the evolution of this behavior. First, through divergence in the thresholds of the receptors expressed at the periphery that are directly sensing environmental temperature. And also in this really unique desert-adapted fly, actually a shift in valence of the signals from the periphery from aversive to attractive, supported by this sort of interesting morphological difference of the key projection neuron that's relaying the signal to the higher brain. Okay, so you may be wondering, as I did, like why be attracted to heat? What is it about adapting to a desert that you need to be attracted to heat? It's somewhat counterintuitive. But when thinking about the thermosensory environment of moavensis, not only is it very hot and dry in the desert, but there are very dramatic shifts and sort of changes in temperature on a daily, seasonal, or even hour-by-hour basis. And so we speculate that heat attraction actually may have originated in the moavensis lineage as a response to the extreme thermal excursions that they experience. So this is actually measurements that I took in the desert when we did sort of a field trip to collect these flies, and you can see that very dramatic changes on a daily basis. And so maybe for this species, actually seeking out heat sort of actively may represent a constant priority. And so they may have actually provided an adaptive advantage to life in the desert. Okay, and so with that, I really want to thank all the amazing people that I did this research with in the Galileo lab, my funding sources, and I really want to quickly share something briefly now. So actually, I just finished my PhD and actually just moved to Harvard last week to start a postdoc in Nick Bologna's lab. And I'm actually establishing this new model system for the evolution of behavior and interspecies interactions by studying these sort of interesting species that establish the foundation for terrestrial ecosystems. So non-vascular plants and these ancient hexapods that are related to insects called springtails. So that's sort of what I'm doing now in the future. And so with that, I'd love to hear your questions.
So questions for Matthew?
Hi, here. Great talk. I'm curious about the humidity sensory neurons, and you show this plot that there's dramatic temperature changes throughout the day for the habitat of Mohavensis, but there's also dramatic changes in the humidity. I was wondering if you see any changes in the PN morphology for the humidity.
Yeah, absolutely. That's definitely, it's something that some people in the Galileo lab are currently doing now to label these projection neurons that carry information about humidity. But what's really interesting too about Mohavensis is they actually prefer really dry conditions. So they like 20% relative humidity. So it could be, we're actively looking at, okay, what are the mechanisms that could drive humidity preference as well in this fly?
Hi, beautiful talk. I was really curious about whether, because these flies live in the desert and because fruit flies prefer a higher temperature during the nighttime, are nights longer in the desert, and do these flies, are they more active in the nighttime, and do they come out in the nighttime, and maybe that's one of the reasons why they may prefer that higher temperature? So did you do this like in different times of the day, night?
No, I didn't do that. We did all the same time of day just for control, but that's an interesting thing to think about. I'm not sure in the desert if there are longer nights. I mean, I'm sure there's a seasonal sort of cycle for that. But actually what we're kind of thinking for the heat attraction is that maybe heat represents sort of rotting cactus, and so that's their food and their egg-laying substrate. So maybe it has to do with like their niche, right? So their food source. But we don't really know what they're doing at night. There's sort of a lack of information about the sort of the ecology of these species. Yeah.
Hi, that was really, really cool. So like thinking about the sister species to Mohavanus, you mentioned that it's also a desert-dwelling species, right? So like,
yeah.
What's kind of going on there that they just don't like the heat?
Yeah. So that's another interesting question. I'm really glad you asked that because it hints at some really interesting species differences. So these species live in the same desert, but they actually occupy very different niches, so like the microenvironment. And so Medleri actually doesn't feed on cactus.
Okay.
Actually, I didn't mention, but actually Mohavensis, if I can switch to the next slide here. No. Mohavensis feeds exclusively on cactus, but Medleri actually feeds on the soil next to the cactus. So like soil that's been soaked with a cactus juice. And that's thought to be a more cooler environment, right? So it sort of maybe represents these differences in their niche.
Awesome, very cool, thank you.
Hi, fantastic talk. I'm just curious on, does the structure of the receptor, the GR28BD receptor itself, is going to contribute to the threshold and like the biochemistry side of this?
Yeah, absolutely. Yeah, that's something really interesting question. So we see that actually there's a lot of sequence divergence in GR28BD when you look at the phylogeny, like a bunch of different Drosophila species. And there are sort of hotspots of sequence divergence in the D exon, so that's the one that's specific to the thermosensory variant, whereas the pore is very conserved. And so we're starting to look at sort of specific parts of the ion channel that could be tuning the threshold. Yeah.
Are there known valence neurons that live in that region of the lateral horn from Drosophila that would provide some logic for why it's going there?
Yeah, so actually we've started to look at, actually now back at the connectome, at the output neurons in the lateral horn. And the number one output of TPN5 in the lateral horn is one we're calling TLON2, thermosensory lateral horn output neuron. And this neuron, if you activate it, it drives avoidance, and it's also, if you silence it, it's required for avoidance behavior. So we think there are different output neurons, and so potentially in Mohavensis, maybe it's rewired to a different set of these output neurons. And there are also, of course, ones that are been characterized that are required for valence of olfactory signals. So, yeah, that's an interesting thing to think about.
Hello, awesome talk. I have a question based on my ignorance of like these comparative genetic approaches. Is the genetic variations for these species different from melanogaster, which I'm assuming is more homogeneous from them being a laboratory strain?
Well, yeah, if you're if you're looking at a laboratory strain, but actually there's quite a lot of different populations of melanogaster. Like if you go to like the ancestral population in Africa, and then they actually followed humans out of Africa, the human migrations, and so you can track the different populations, and there's quite a lot of difference even within species. And Mohavensis actually has 4 different subpopulations very distinct populations. And so I actually looked at the behavior of those 4 different ones, and they all have this heat attraction phenotype. So yeah, there definitely is interesting population level differences, but at least this behavior seems stable at the species level. Yeah.
Hi, fantastic talk. I was just curious, with these different temperature preferences, do they have differences in lifespan?
Oh yeah, they do. Well, kind of like how if you can raise melanogaster at different temperatures, they actually live longer. Moavensis, if you raise them at just room temperature, they live much longer, and they're very lethargic and slow because they're used to the heat. But so if you raise them at like 30 degrees, 35 degrees, it's roughly the same lifespan, but they take much longer to reach sexual maturity.
Actually, kind of like related to what you just mentioned, can you change kind of like the preference phenotype based on your culturing temperature? Like if you were to move them for X amount of generations, does that change the preference?
Yeah, so for this this behavior, it seems to be very innate. So it doesn't matter like what temperature you raise them at. They always, like melanogaster always prefers 25. There may be some slight differences in like the degree of avoidance, but their preferred temperature seems to be a very innate behavior.
Over here, Matt.
Hi, yeah, great talk. I'm wondering, is the reproduction tied to these rotten cactuses? Since you mentioned that they're around 40 degrees, and could that be the drive for the switch in temperature? Because they have to get to where they're mating?
Yeah, that's a good point. Yeah, I think that you could be getting onto something, because heat attraction is quite different from heat avoidance, right? Kind of what Yoshiaki, I think, talked about earlier. So it seems that heat attraction has the advantage, right, where they're actively seeking out and heat. So maybe there's something to do with what they're eating. It helps them find cactus, because obviously it's not required for adaptation to the desert, because this other fly still avoids heat, right? So.
Hi.
Yeah.
Great talk. Forgive my ignorance, but is it known or studied if there are any major metabolic differences in these sister species that could have evolved first and then drive this rewiring in the lateral horn?
Yeah, so you're getting at a question that's really interesting that I've thought about a lot, is that the interplay between preference and tolerance, right? So a species needs to tolerate a certain temperature, and they also need, they have a certain preference for temperature, but it's unclear like how those co-evolve, and maybe you could have one without the other. I mean, obviously it wouldn't be good for a polar bear to like hot temperature, but yeah, I mean, that would be interesting to look at, like along the phylogeny, maybe at a more granular scale, like look at the species, and maybe some species can tolerate heat but doesn't have the preference, and so maybe you could get at that question of like how those two co-evolve. Yeah.
Hi, great talk. Just a simple question. Do you see different isoforms of GR2A? And do you see like some species have certain sort of isoform versus, like, is there a nice demarcation between the type of isoforms each?
Yeah, so actually this thermosensory isoform is specific to flies, so it's only found in Diptera. And actually it's absent in mosquitoes, so it seems like, and actually if you look into the mosquito genomes, you can see they only have 4 isoforms, 4 exons there. So it seems like this duplication event occurred somewhere in between mosquitoes and Drosophila, so somewhere like in that phylogeny.
Okay, if there are no other questions, there's one more actually, sorry.
Okay, thank you. Great talk. So I wonder, when they sense the heat, they avoid, right? What's the locomotion strategy they use, like in terms of increasing like forward speed or turn, or do they like use like gradient information between 2 antennae, or just the absolute temperature?
Yeah. There's actually some published work from the lab from a couple years ago where we're looking at sort of how the flies navigate this sort of similar gradient that they experience in the assay. So the flies actually can use the difference between temperature and their antennae in a very a very stark gradient to be able to like turn away from heat. And so they actually do use their antennae in that way, as sort of as they're going upwards in the gradient, then they can turn to avoid. But they may also use other mechanisms that are independent of like the difference between the antenna, right? That's more of like a temporal sort of basis for the behavior.
Beautiful talk. I'm wondering if you know anything about the larvae of these different species and if they express the same ion channel with different sensitivities or anything.
Yeah, I tend to forget about the larvae, to be honest. They're kind of like a different organism in a way, like very, very different sort of lifestyle. Because they kind of just stay within the food patches that the adults lay in there. So there are like many labs that are looking at temperature preference in larvae, and they actually do have distinct preferences. I think for melanogaster, it's actually like more 24 degrees is their preferred temperature, but very close. But I don't know about GR28BD actually. I don't think don't know where it's expressed in larvae.
Okay, so there are no other questions. We're going to come back at 8:30 with a sensory session, and let's thank Matthew one more time for a great talk.